So I really just started the lecture of the retina and beyond, as I call it. This is the second part. And I started with this concept of a neuron's receptive field. I'm just going to repeat this because it really is an important concept. If you're going on in sensation and perception, if you're going on in this particular field, it's important. But it's also important if you're going on in medicine or physical therapy or neurology, that the neurons in the brain that are responding to sensory information, they are responding to specific receptors. Okay, so the area on the receptors that is influencing the firing rate of a neuron is part of that neuron's receptive field. And now that we've talked about excitatory versus inhibitory effects, and we've talked a little bit about lateral inhibition, whether I'm having an excitatory influence on the neuron or an inhibitory influence on the neuron, I am part of that neuron's receptive field. And these neurons have these receptive fields for the neurons have different sizes and shapes and properties. And we started with the really simple retinal ganglion cells have that center surround receptive field. And one thing I know I did leave off when I was talking was I talked about the excitatory center, inhibitory surround receptive fields, but we also do see inhibitory center and excitatory surround receptive fields, both in the retinal ganglion cells and in the lateral geniculate nucleus, which I'm pretty sure I'm going to be able to introduce during this presentation. So I really just wanted to have a slide up here to come back to this concept of lateral inhibition. I described how we discovered it, a little bit about what it does for us. I did have some people at the end of class asking questions that I will come back to in one of the Monday classes. I'm not sure I'm going to have time next Monday, so it might be during our review where I try to really explain how lateral inhibition gives us the contrasts and the changes in our environment, how it spotlights those and makes those more salient for us, which is really what we want to see. For seeing the shapes of things, I want to see the edge of my desk or my table so that when I put my drink down, I put it in the middle and not where it's going to fall off. So lateral inhibition is doing that for us. And we also went through the Hermann grid and the kind of proof of how lateral inhibition is influencing, how that is causing us to see those gray ghosts. Because really, what we want to see and what we see with our visual system is relative light that is in colors that are relative and influenced by what's around them. I'm going to use another illusion to show how lateral inhibition is influencing our visual system. I'm going to use simultaneous contrast. Your author uses the Chevreul illusion, which I'm used to calling Mach bands. I think they are slightly different from each other, but they're a very similar kind of illusion. But he uses numbers again, and I like to give one example that doesn't use numbers, because some people have just a lot of anxiety whenever numbers come into the scene. They think, oh no, math, numbers, and this is a nice example that I think he used to have in there that doesn't use numbers. So when I do this in class, I ask, and so you can think about if you're looking at the inner gray squares in this picture, which one appears darker to you, the one on the left or the one on the right? And when I say appears, most of us say the one on the left appears slightly darker than the one on the right, although I do have some students this does not work on. And you probably know the punchline, but maybe you don't. The two inner squares, they are the same, they're giving us the same exact wavelength information. The same exact information is being reflected into your eye in both of those, from both of those inner squares. And what's happening here is the one on the left has a lighter background that it is on, and so that lighter background is sending more inhibition laterally, causing that middle square to appear darker. The one on the right has a darker background, and so it's sending less inhibition. It's sending less light, so less inhibition laterally along that same path, and so that makes that inner square appear to be a bit lighter than the one on the left. And because he gives us this example in the book, or he used to, with the big arrows versus small arrows, I just think it's helpful. And so you can go back and look at that again. It really is, I don't know if you believe me, that the one on the left and the one on the right are sending the same wavelength information. And the idea is, on the left, that light background is sending more inhibition, which here is being denoted with the wider arrows. The one on the right, that dark background, is sending less inhibition, and so that one appears to be lighter than the one on the left. If anybody has questions about Mach bands or the Chevreul illusion, or he actually has, I'm about to go into an example of an illusion that for which lateral inhibition does not work to describe the illusion, and he goes through a couple of those. So if you want to look at those in the book for different examples, if you think it'll help and you have any questions, just let me know. And now I'm going to talk about White's illusion, which lateral inhibition cannot explain. And you'll notice that there's actually, I think your book now has a different rendition of the Hermann grid, where you don't see those gray ghosts, and which lateral inhibition also cannot explain why. Why don't you see those gray ghosts in the second example? But just to show that it's not all easily explained by lateral inhibition and what's going on at the retina, at the level of the retina. Sometimes we have to take into account what's happening at the visual cortex. And so if we look at White's illusion, most of us have this experience. If I asked you which gray rectangle appears darker, the one on the left, A, or the one on the right, B, most of us have this experience that A appears to be darker than B. And I'm again going to tell you it's the exact same, they're the exact same shade of gray, same wavelength information being sent to the eyes. So what's different about A and B is the context. But if you look at what's around A is mostly surrounded by those dark bands, right? Which would lead, lateral inhibition would mean A would be lighter. And that's not what happens. So that's annoying. And so White's illusion is that he explains that context is really important, and that we're interpreting that gray rectangle as being on a white background with black stripes coming through it. And so that larger kind of context is brought to bear. And lateral explanation cannot easily explain that. We do have neurons that are have excitatory and inhibitory kinds of influences on our color vision up at the visual cortex, but so we have to consider this kind of larger interpretation, even though what we just explained was lateral inhibition at the level of the retina. That really does not explain White's illusion at all. I'll go ahead and walk through B. So if you notice, there's more white areas hitting that gray than in A. It should have more lateral inhibition. It should appear darker. That's not what happens. And the assumption is that we are interpreting that gray rectangle as being on a black background with white stripes going through it. That one doesn't work as well for me, but for some of you it might. It's hard for me to see the black background. It just looks like a weird illusion. But A works very well for me as feeling like the context is that that's on a white background. And so our interpretation of context is influencing our interpretation of the gray squares themselves. Okay, so the final kind of what you really need to know is this, and I have it all on one slide. This is what you can memorize. If everything I just said and all those proofs just annoy you and you don't really understand, it's okay. Just remember, lateral inhibition refers to the fact that an excited neuron may reduce the activity of its neighbors. This means that if a single neuron is activated, it's going to respond more strongly than if its neighbors are also stimulated, because they're often sending inhibition to neurons along the same path. Lateral inhibition increases our ability to see contrasts and changes in our environment. So this kind of relative light and seeing when things change. And I usually do this explanation with the center surround receptive fields going across a light-dark kind of change. And if anybody wants me to do that on Monday, I think it will be tricky in Blackboard Collaborate, but I'll figure out a way to, I'll at least think it through. It might take some time, but I'll think it through. Okay, we're going to get to our primary visual pathways, getting out of the retina. And first I'm going to do this, how did we get here? Which is an allusion to the Talking Heads, just so you know, of that song, How Did I Get Here? So light reflected from an object into the eye, light focused to form an image of that object on the retina. That light in a pattern that illuminated some receptors intensely, others more dimly, is absorbed by those visual pigment molecules that are packed in the rods and the cones in their outer segments. There are chemical reactions in the outer segments of those photoreceptors, and they transduce the light into electrical signals. As these electrical signals travel through the retina, because we have those photoreceptors, bipolar cells, retinal ganglion cells, and between those two, the horizontal and amacrine cells doing the crosstalk, those electrical signals interact, they excite and inhibit each other. The electrical signals reach the retinal ganglion cells, which because of this processing have center surround receptive fields. Remember, some are excitatory center inhibitory surround, some are inhibitory center excitatory surround. After being processed by the retina, these signals are sent out of the back of the eye in the optic nerve, which this is made up of the nerve fibers, those axons, of the retinal ganglion cells. Okay, so we're here at the optic nerve, and we're going to send that information along some primary visual pathways. First of all, it gets split between the lateral geniculate nucleus and the superior colliculus. I'm going to ignore the kind of what's going to the hypothalamus, but so the majority of signals are going out through the optic nerve, and they are reaching the lateral geniculate nucleus of the thalamus. If you know what the thalamus does, it's really the kind of way station for sensory information. It is organizing, summarizing that information to send it on for higher level processing. And for the vision, that's happening in the lateral geniculate nucleus. Approximately 10% of the signals go to the superior colliculus. So the superior colliculi, we have one in each hemisphere of the brain. They are subcortical structures that are controlling eye movements and other visual behaviors. If you remember me talking early on about the complexity of perception and how difficult it is for us to create a machine that can even navigate its environment. And when we do this with cars, we often use lots and lots of cameras as well as sonar or some other kind of information about where things are, because we cannot get those cameras to kind of move as we have eye movements that are based on what is relevant to me at this time. The superior colliculus also takes in information from other modalities to help control eye movements. As far as, so what goes, most of the information goes to the lateral geniculate nucleus and is sent along then for higher level processing to the primary visual cortex. And I have here the primary visual cortex, the striate cortex. So striate means striped. It has this kind of striped appearance, and they sometimes call it that. Sometimes called V1, meaning primary visual cortex, because just different articles that you're going to read are going to use different nomenclature. From the primary visual cortex, we're going to pass that information along to two pathways, to the temporal lobes for object recognition and to the parietal lobes to help us to navigate in space. One of the aspects of the way visual information is sent to and through the brain is that we have contralaterality. So if you fixate straight ahead, I'm just going to use this example. So if I'm looking straight ahead at a fixation cross in the middle and there's a tree to my left and a house to my right, the left half of each eye is going to receive images from the right visual field. So the left half of each eye is getting that kind of flipped, inverted image of the house. Because remember, I'm looking straight ahead, so it's hitting the left retinas and going back to the left hemisphere. If I'm looking straight ahead and the tree is off to the left, that part of the image, because again it's flipped and upside down, that it is hitting the right half of each retina. And so the right half of each retina is going to send that information back to the right hemisphere. And what you can see is for the kind of outsides of the retina, I'm sending that back ipsilateral. That's to the same side, to the same hemisphere. But for the insides of each retina, I have to send that contralateral. I have to cross hemispheres, and we see that happening at this optic chiasm. So the optic chiasm is right where that X is, where the solid line is passing through the dashed line. And we have, for vision, this kind of complicated contralaterality because it's about the visual field. The lateral geniculate nucleus, again, this is a subcortical structure. It's in the thalamus. Its function is to control the flow of information, to organize information. It's doing some summarizing of this information. I often think of the thalamus and these nuclei as little librarians for us. It's getting input, and I say here at least from what we understand, it's getting that input from the retina, but it's also getting input from the other nuclei in the thalamus, from the visual cortex, and this kind of top-down processing. The visual cortex is communicating back from itself, from within the lateral geniculate nucleus, and from the brainstem. So when something's really important, I hear a loud noise or something, I would need to move my eyes, or I need to really pay attention and set down those features, that is also input. Whereas its output is primarily to the visual cortex, okay, primarily to the cortex. So we have this 4:1 ratio of afferents to efferents, where afferents are arriving fibers, or the input, and efferents are exiting fibers, or the output. And it's doing a lot of summarizing of information. Signals arriving to the lateral geniculate nucleus are sorted and organized based on a number of things. One, the eye they came from. Two, the specific kinds of receptors that generated them, so whether it was rods or cones. And then also the type of environmental information they represent. We won't talk a lot about this again, but color information is separate from movement information, all of that in the lateral geniculate nucleus. We've pulled that kind of apart, and we have to put it back together. So one aspect of the lateral geniculate nucleus's organization is that it has this retinotopic map. So A is closer to B than it is to C, both on the cup and saucer and what we see in the retina, right? Because what's up at A is going to hit down at the A prime, or the lower part of the retina. B kind of straight back. C is going to go up. And again, B is closer to C and B is closer to A than A is to C. A and C are far apart. And if we look in the lateral geniculate nucleus, we have maintained this retinotopic map. A is close to B, B is close to C, A and C are far apart. So moving from that picture where A, B, and C are along the cup and saucer, A and C are farther apart, we're going to go ahead and add D here. A and D are even farther apart. A, B, C, and D are points on the retina, basically. And we have this retinotopic map where we have columns that correspond to those particular retinal points, okay? And you can see that we have 6 layers in the lateral geniculate nucleus. We're not going to talk about this very much in this class, but if you take brain and behavior or neuroscience or something, the cerebral cortex, we also see these 6 layers of information, where different information is being kept in those different layers. Okay, and this is kind of a more simplistic map, and I'm going to show another one in a minute. But as we're looking at those columns that are corresponding to particular retinal points, we can see also that we have the blue and the red. The blue, so the rows of 1, 4, and 6, as we kind of shuffle this information in together, that is the contralateral input, so the input from the contralateral eye. And the red, rows 2, 3, and 5, is the ipsilateral input, or the input from the ipsilateral eye, the eye on the same side of the brain. And again, those are amazingly, due to an amazing feat during our neural development, those are shuffled together perfectly so that they match up, and we can have these corresponding points from the retina matching up and meeting each other in the lateral geniculate nucleus. So moving on to the superior colliculus, its function is, overall, what it's doing is it's allowing for eye movement control, and so localization in space is really important. It has these spatiotopic maps, and it is receiving information from different modalities, so different senses. So it also has this kind of multimodal spatial coordination. It's really quite impressive, and again, we really don't understand a lot of how the superior colliculus is allowing us this extra information of our eye movement and letting the brain know where our eyes are looking and putting it all together with other information so that we know where to move our eyes.